Good morning. In this lecture series, we have to see about the other type of measurement techniques. Okay, it's like a hemoglobin measurement techniques. Let's move on the topic of the hemoglobin, how it can be measured and how it can be flow. We have to follow that. The first of all, the hemoglobin measurement is the techniques. Okay, so how it can be DT empathize here. The hemoglobin is a measurement is a which way is vital the assessing the oxygen transportation in the blood and the various techniques exists and with each specific applications and accuracy level. This presentation is to summarize the primary methods and their significance in the clinical settings. So the hemoglobin is nothing but the protein in the red blood cells and responsible for the transporting oxygen from the lungs to tissues and returning the carbon dioxide for the exhale. Okay, the important measurements are the accurate the hemoglobin measurements is essential the diagnosing anemia is a polycythemia and other blood disorders. So for example, the hemoglobin can be transferred as the oxygen transfer and carbon dioxide. So here we have to represent that. So both we have to different multiple colors we have to represent. That is red color is can be shows as the hemoglobin. That is the central protein in the red blood cell. So the oxygen transfer is represented as the blue color. It can be delivered the oxygen from the lungs to the tissues and the carbon dioxide transport is represented as the green color and is facilitates the carbon dioxide is written for the exhalations. So what are the spectrometry of the overview? The what are the basic principle of this is measuring the absorptions of the light by the hemoglobin and correlating with the concentration. The common methods are we have to including the co-parametrics and it's a differential hemoglobin forms and enhancing the accuracy. So these are the advantages for the using the spectrometry of the overview of this the non-invasive and quick result and suitable for the point of the carrying testing. So the hemoglobin entropiasis can be carrying the three or four six types of method. That is the first way we have to calculate the prepare the sample and applying the electrical field and this hemoglobin mitigations and identify the variants and diagnose the disorders and managing the treatment. So these are the flow pipe can be using for the hemoentropias. So the preparing the sample is a basic the to collect this blood sample for the purpose of the analyzing. So after this process can be applying the how the electrical fields can be applied to the gel. So then is a more this third stage is a hemoglobin variance is a mitigate the different types of rate and the hemoglobin variance is are identified the based on the mobility and is a disorder diagnosing the disorders. The results are used to diagnosing the hemoglobin neopsias and is a clinical using the result to managing the patient treatment based upon the managing treatment. So the hemoglobin A1C testing is a diabetes the monitoring testing method and a clinical significance. The hemoglobin A1C testing is a measure the average blood glucose the level over the past 2 or 3 months. The common methods are including the higher performance liquid, the chromatography that is the hemolysis and the regular the A1 test helps to treatment the adjustment and the risk assessment for the complications. So the point of carrying the testing, so there are three types out there that is a rapid diagnosing tool and types of device and advantage and challenges. So what is the rapid diagnosis tool? Is a point of care testing device to allow the immediate hemoglobin measurement. So the types of devices are these including the analyzer. Okay, how is can be handle the device. Okay, that's analyzing the utilizing the how the measurement techniques. So what are the advantages and challenges here? The while offering the rapid results, the accuracy of the point of care test can be vary and necessitations and confirmations with the laboratory methods in the critical situations. So the conclusion of the hemoglobin measurement techniques. So there are diverse techniques and future developments and implications for the healthcare. So the variety of allow the trials and approaches the enhancing the patient care. The future's developments are the research focusing on the accuracy, speed, and accessibility through the innovations. The implications for healthcare, essential understanding for the informed decisions in the treatment blood disorders. So the let's summary of this case. The hemoglobin is a measurement techniques. Okay, as we know that the significant clinic setting and technique characteristics and focusing on the summary. So as we know that the significant clinic statistic and the technical characteristics are the various techniques are exhibiting the specific applications and different accuracy levels. And so focusing of the summarize the primary method and the clinical significance. So these are the techniques we have to using for the hemoglobin measurement techniques. Thank you.